This is lecture 5.2 on cellular reproduction. In this lecture, you're going to learn about the cell cycle, explain how DNA forms chromosomes, name and explain the stages of mitosis, name and explain the stages of meiosis, describe the importance of meiosis for genetic recombination, and describe the consequences of non-disjunction for human health. So I know what you're thinking. Yay, mitosis and meiosis. Not. For many students, this is one of the most boring topics and they absolutely get through it because they have to. However, I would argue that if you don't understand what's going on in mitosis and meiosis, it's going to be very difficult to understand the basic premises of Mendelian genetics. And this is the point in the semester where I tell you what my evil plan is. If you looked at the schedule for the semester, you'll notice that later in the semester we're going to be talking about evolutionary biology. And for many people, evolution is a topic that is a bit nerve-wracking. They don't really understand it. And the mechanisms behind it seem to be somewhat magical. However, this is science class, not magic class. And I think the main problem is that it would be a bit like me taking you and throwing you into an advanced calculus class without training you on the intermediate steps. So if you've taken all of the intermediate classes and passed them, calculus is just going to be the logical next step. So I'm going to help you to get to the point where the later lectures are going to make perfect sense. And to do that, we need to start with something quite fundamental, and that is cell reproduction. One of the foundations of modern biology is cell theory, and that is all cells come from pre-existing cells, except for in the very beginning. And how those cells make new cells has got to do with cellular reproduction. Now, if you study bacteria, you'll learn about fission, but we're going to be focusing on eukaryotic cells, specifically animal cells, and we'll learn about two main ways in which cells reproduce themselves, and we'll compare and contrast those methods. The first one is called mitosis, and I jokingly say the way to remember mitosis versus meiosis is that mitosis can happen in my toes when I cut my foot, whereas meiosis happens in my O's, my ovaries, or my gonads. So there are 4 stages to mitosis: prophase, metaphase, telophase, and anaphase. You can use a rhyme to remember it. We often have our students, oh, look, did I just do this wrong? PMAT. I've actually got this slide incorrect. I will have to fix that. It should be prophase, metaphase, anaphase, telophase. That was a great error for me to have in there. I'll fix it in your slides. Remember that DNA is actually replicated in the S phase. That's not part of mitosis. So the cell will actually enter prophase with 2 copies of every strand of DNA. So if we think back to that human karyotype, you had 46 chromosomes. You had 23 pairs of chromosomes. But that's a cell in G1. So a cell that's entering mitosis would actually have, drum roll, twice as much. It would have 46 plus 46. Okay. The function of mitosis is to make sure that when you make a new cell, you have the right amount of DNA in each new cell. If I'm making a new cell to fix my toe that's got a cut on it, I need to make a new skin cell. And my new skin cell has to be a diploid cell. So I need to make sure that I double my amount of DNA and then I go through 1 round of division to then get it back to what it was before. What we're going to do next is describe briefly some of the primary characteristics that you would see in each one of those 4 phases of mitosis. So if we start out in prophase, what's going to be going on? Well, at this point in time, DNA starts to wind around histones and form chromosomes. It didn't do that before because Before you enter mitosis, the cell needed to actually use the DNA, so it needed the DNA to be unpackaged so that the cell's machinery could actually attach to the DNA and copy it and use it. But when you're getting ready for mitosis, you got to get ready for that journey. You've got to pack your bags. And part of that packing is the formation of chromosomes. So when you see a cell that's entering prophase, what you would notice when you look at slides under the microscope is that you start to see these clumpy sausage-like structures. Those are the chromosomes forming. Now, the chromosomes don't just move themselves, they need to have something to move them. There are special little fibers, part of the intracellular skeleton, that forms these little spindle fibers, and those emerge from structures called the centrosomes. The centrosomes kind of spin a web through the cell, creating tracks that the chromosomes can attach to and move on. Now, normally the DNA is kept within the nucleus, and that's that double membrane layer. But it's hard to move things if they're still packaged in that nuclear envelope. So the nuclear envelope actually has to break down so that the DNA can spill out of it and move around. The centrosomes move to opposite sides of the cell so that they can give an orientation to where each new cell should be. Metaphase is one of the phases that's easy to recognize. Meta means middle, and it kind of both represents the middle of this process as well as the chromosomes physically being in the middle of the cell. The chromosomes are consisting of pairs of chromatids that are identical to each other and connected at the center of them. Those attach to the spindle, and they're attaching along a midline called the metaphase plate. The very next part of that process is that those sister chromatids get pulled apart or popped apart and get moved along the tracks to opposite sides of the cell. Anaphase is that process between the cells being at metaphase in the middle and having been pulled to opposite sides of the cell. You can often recognize a cell that's in anaphase because the chromosomes have a V-like structure to them. If you took a piece of spaghetti by the middle, cooked spaghetti, not raw, if you took a piece of cooked spaghetti by the middle and you pulled it along a surface, what you would notice is where you're pinching it would form the apex of a V, and then the two long ends of your spaghetti would drift behind it. So you could actually see which direction you're pulling your noodle in, depending on which direction the V is pointing in. And in anaphase, you can generally tell that the chromosomes inside of the cell are being pulled to opposite ends. Telo means long, like telephone is to call someone a long way away. Telo is the end, the very end of the mitotic process. At this point in time, the chromosomes have reached the opposite poles and they stop being so tightly wound up. You can start unpackaging a little bit. Nuclear envelopes start to form around each new nucleus, and the mitotic spindle starts to break down because it's not needed anymore. After telophase, you can get a process called cytokinesis, and that means the breaking of the two cells into two separate cells. And this happens a little differently in animal cells versus plant cells. Cytokinesis is not considered part of mitosis, generally happens afterwards. So, if you think you know all about mitosis, I do recommend that you go back into the lecture and you click on this mitosis self-quiz and see if you get the answers right. All right, I'm going to press pause on this. Oh no, I'm not, I'll let it go. Sexual reproduction and meiosis. So Meiosis is the way in which you would make gamete cells, so either eggs or sperm, and they're haploid. This particular video that I pulled is human sperm under the microscope, and they have flagella, which is kind of cool, but each one of them is haploid. It only has 23 chromosomes, one copy of the genome. So making those is going to be a little bit more complicated. So in humans, eggs and sperm are haploid, or 1N, and when the egg is fertilized, it makes a diploid zygote. And that can start to develop via the process of mitosis into a new human. There are special names for meiosis in humans. The production of sperm cells is called spermatogenesis, and it happens in the testes of a male. It starts when you enter puberty, it's continuous, and it ends at death. Oogenesis is the making of ova, or egg cells, and that happens in ovaries of females. This starts at puberty, and ovulation occurs once a month. So you go through the first part of meiosis once a month, and it ends at menopause at around age 50. So just to sort of remind you that human adult karyotype, 23 pairs of homologous chromosomes. Now the karyotype that I've put in this picture looks a little different from the previous one. This is a diagrammatic, and it shows these bands that are light and dark. That corresponds to actual bands of lightness and darkness on those chromosomes. It's how they are lined up with each other. And where you find that dotted line, that's the center of each chromosome where they attach to each other. So you get one homologue from mom and one homologue from dad. Homo means the same as. So homologous chromosomes are basically chromosomes that have got the exact same set of genes on them. Now, the genes don't have to be identical. I like to think about them as recipes. So you've got two recipe books, and they both have recipes for brownies. But one is your mom's recipe for brownies and one's your dad's recipe for brownies. And they might result in slightly different products. Now, the goal of meiosis is to have only one copy of each of the homologous pairs. Now, which copy you get, whether you get your mom's copy of chromosome number 1 or your dad's copy of chromosome number 1, is up to random chance. We'll talk about that in a little bit. But so for each one of those 23 pairs of chromosomes, you're going to select one of those. So during the S phase, each chromosome duplicates. So you end up with 2 copies of each pair of homologous chromosomes. So what that means is that when you get into G2 and you're about to enter meiosis, how many copies of chromosome number 1 would you have? Would you have 2 pairs? Nope. You would actually end up with 4 copies. 2 of those are copies of your mother's, and 2 of them are copies from your father. So you would end up with 4 chromosomes, and that number is important. Now, your goal is to end up getting a cell that's only got 1 copy. So if you're entering meiosis with 4 copies, and your goal is to get down to 1 copy, that means you have to have 2 rounds of division. So meiosis goes through its process twice. We have meiosis 1, in which a 4N cell is divided into 2 2N cells. And then there's meiosis 2, in which each one of those 2 2N cells is divided into a 1N cell, with a result of 4 haploid cells. Meiosis 1 has got prophase, metaphase, anaphase, and telophase. There is no prophase 2. The cell immediately goes into metaphase 2, anaphase 2, and telophase 2. And prophase 1 is particularly important. There's interesting stuff happening here. And the stuff that's interesting here is specifically interesting because it relates to genetics and inheritance, and that specifically relates to evolutionary biology. So the first thing to pay attention to is something called tetrads. Now remember I told you that there ends up being 4 copies of chromosome number 1. Tetra means 4. All 4 of those chromatids, the 2 pairs of homologous chromosomes, line up with each other. And when they line up with each other, something funny can happen, and it's called crossing over. Those long pieces of DNA can lay over each other to form a crossing where they actually exchange segments of DNA with each other. And what this does is it creates hybrid chromosomes. It creates a chromosome where you have some genes that came from the mom mixed up with some genes that came from the dad. So the offspring is not going to have just a pure maternal chromosome number 1 or a pure paternal chromosome number 1, but it could have a combination of both of those. The second thing that happens, and this is during metaphase 1 and metaphase 2, is a process called independent assortment. Independent assortment is going to be fundamental to understanding one of your laws of Mendelian inheritance. What happens at metaphase is that the chromosomes line up along that metaphase plate. But how they line up is random. Whether they line up with the maternal chromosome on the left-hand side or the paternal chromosome on the right is at random. And this happens not once, but twice. So you get shuffling of genes because you could get your mom's version of chromosome 1 and your dad's version of chromosome 2. Or you could get your mom's version of chromosome 1 and your mom's chromosome 2. Which one you get, that's a flip of the coin. Or rather, a flip of the chromosomes, because how they line up at that metaphase plate is at random. So these two processes result in the formation of gametes that are different from each other and different from the parent. So let's have a quick look at meiosis in its sequence. So during prophase 1, just like in mitosis, you get the chromosome condensing, the nuclear envelope breaks down, but during prophase 1, bing bong, this is where you get tetrads and crossing over. During metaphase 1, you get the pairs of homologous chromosomes. So you have 4 chromosomes, so it looks like 2 butterflies next to each other at that midline. At metaphase 1, those 2 pairs of homologs break apart from each other and move to opposite poles of the cell. At the end of meiosis 1, you have chromosomes that still have 2 chromatids in each chromosome, or 2 strands of DNA in each chromosome. You immediately enter meiosis 2. And I did tell you that there's really no prophase 2. In some books, you'll see it being represented, but basically your cell goes straight into the next part of that process, where the chromosomes now line up against a metaphase plate that is at 90 degrees from the previous one. This time, the chromosomes line up and the sister chromatids separate from each other and moving to opposite poles of the cells. Now bear in mind that this is not just happening in one cell, but it's happening in 2 cells. So you end up at the very end of meiosis 2 with a total of 4 cells forming. Meiosis can sometimes go wrong. What if you don't end up with separation during metaphase? What if the chromosomes don't pull apart properly? That's called non-disjunction, and it results in the wrong number of chromosomes being in the daughter cell. There are quite a few human chromosomal disorders that are caused by non-disjunction, and these include chromosomal disorders where there is non-disjunction in the autosomes. So for example, if you end up with 3 copies of the 21st chromosome, that would result in a disorder referred to as Down syndrome. Down syndrome is compatible with life, but people with Down syndrome sometimes have much shorter lives and they have a cognitive impairment, heart dysfunctions, and some other disorders. Trisomy 18 and trisomy 13 are trisomies that are not compatible with life. And what that means is that if an embryo has got trisomy 18 or 13, that child is likely to either die in utero or very shortly after birth, and it will not be able to grow up. Sex chromosomes can also have monosomy or trisomy. Klinefelter syndrome is where there is an extra copy of the X chromosome. Now, in normal females that have 2 copies of the X chromosome, we don't actually use both of those copies of the X chromosome. The body takes one of them and scrunches it up like a piece of paper into something called a bar body. And it only uses one of those copies of the X chromosome. So in a person who has Klinefelter syndrome, one of their X chromosomes will be scrunched up and not used. And so they'll end up fundamentally having a chromosome set that mimics that of a male. A Klinefelter person is typically considered male because of the Y chromosome. The presence of a Y chromosome confers maleness onto a person. Trisomy X is where there are 3 copies of the X chromosome. In that case, typically you'd end up with 2 bar bodies forming in each cell. Turner syndrome is called monosomy, and that is where you only have an X chromosome. You don't have an extra Y chromosome or an X chromosome. And so in that case, you would not form any bar bodies in the cell, otherwise you wouldn't be able to have a functioning X chromosome. The X chromosome has got genes that are used by both males and females in their body. A person with Turner syndrome is going to be female, but is going to have some levels of infertility and some other symptoms as well. Now, non-disjunction is something that happens at random during meiosis, and it can happen in both sperm and in eggs. Now, in human females, the rate of non-disjunction errors increases with maternal age. After about age 35, the rates of non-disjunction go up very rapidly, and that's one of the reasons why women are very carefully monitored if they have pregnancies over the age of 35. And there are various different options for being able to test to see if your fetus has a normal chromosome karyotype or not very early in pregnancy.